A very good day to each and every one. I welcome you all once again to part 10 of chapter 1 Reproduction in Lower and Higher Plants, class 12 Biology, revised syllabus 2020 of Maharashtra State Board, which also covers NCERT Unit VI Chapter 2. And I am your facilitator Nusrat Siddiqui to help you and to guide you, having experience of more than 15 years in the field of teaching. Let's begin our video with yet another inspiring quote by Anthony J. Develop a passion for learning. If you do, you will never cease to grow. Since each new day teaches us new things, whether we realize it or not. This process is mostly unconscious. That is why we are mostly unaware. In order to develop our passion for it, we should learn how to ask questions. Once we have learned that, we should try and give them an answer. Once we know the answer, we should teach others. The infinite satisfaction you get makes your passion for learning grow bigger, and doing it repeatedly makes you grow and grow wiser. So let's grow wiser and start learning. Let us do a quick recap as to what we have covered and studied in our previous videos. So we have discussed about structure of flower, structure of anther, structure of anatropous ovule. Then we have also studied about process of microsporogenesis and megasporogenesis. We have learnt about pollination and its type. We have also understood outbreeding devices and how the plants or the flowers, they develop the devices in order to promote cross pollination. So this is what we have studied up till now. Today we are going to discuss about pollen-pistil interaction. It begins with pollination and ends with fertilization. We are also going to discuss about artificial hybridization. Let us understand what is pollen-pistil interaction. Basically, it is an interaction between the pollen grain and stigma. It begins with pollination. We know what happens in pollination. In pollination, the pollen grains lands onto the stigma. So it begins with pollination and it ends with fertilization. And all the events, right from the pollen grain getting deposited onto the stigma to the entry of pollen tube into the ovule, all these events comes under or are a part of pollen-pistil interaction. And we all know that pollen tubes germinate from the germ pore. We know that pollination does not guarantee the transfer of right type of pollen. Often the wrong type also lands onto the stigma. Therefore, pistil has the ability to recognize and accept the right and compatible pollen grain of the same species. Thus, the wrong type of the pollen is discarded by the pistil. Now, the compatibility and incompatibility of the pollen grain and the pistil is determined by the special proteins. This process involves pollen recognition, that is, pollen is recognized whether it is compatible pollen, and if it is a compatible pollen, then it is followed by the promotion of that compatible pollen, that is, it is allowed to germinate. And if the pollen is incompatible or the wrong pollen, then it is inhibited. The growth of that pollen is inhibited. The growth of incompatible pollen is inhibited. It will not allow, the pistil will not allow it to germinate. The right pollen or the compatible pollen will absorb water and nutrients. Nutrients, mainly sugar, from the surface of the stigma. Okay, and thus it will produce the pollen tube. So from the surface of the stigma, it absorbs the nourishment, it absorbs the nutrients, mainly sugar, and then this is absorption is done by the pollen grain through the germ pore, and it germinates and produces pollen tube, and this pollen tube grows through the style, and this growth is determined by the specific chemical. Thus, the stigma, it provides a essential prerequisite for successful germination of the pollen tube, which is absent in the pollen grain. We know that plants shed pollen grains at two-cell stage. Two-cell stage means a vegetative cell and a generative cell. So in a pollen grain, mostly the pollen grains are shed at two-cell stage. The bigger one will be the vegetative cell and the smaller one is the generative cell. And after a compatible pollination, means when the right type of pollen is deposited onto the stigma, then the pollen tube is produced through the germ pores, and after this, when the pollen tube is produced through the germ pore, the content of the pollen moves into the pollen tube, and over here the generative cell, it undergoes one mitotic cell division and forms two male gamete during the growth of the pollen tube. So the generative cell, it undergoes one mitotic cell division and then it forms two male gamete. So this is the two male gamete which is formed after the formation of or during the formation of pollen tube. And in plants which sheds pollen in 3-cell stage, that means it already has two male gametes from the beginning, then in that case the pollen tube will carry two male gametes from the very beginning. Let us understand some features of the pollen tube. Boron and calcium are the ions which are essential for the growth of pollen tube. Mainly boron helps in the growth of pollen tube. The ideal temperature or the best temperature for the growth of pollen tube is 20 degree to 30 degree Celsius, and pollen tube shows apical growth and chemotropic movement. Now in case of pollen tube, generally one pollen tube arises from the pollen grain, and this is termed as Monosiphonous, which is most common one. So generally mostly only one pollen tube arises from the pollen grain, which is most common. But if there are many pollen tube arising from the pollen grain, then it is termed as Polysiphonous. Now even though there are many pollen tube arising from all the germ pore or many germ pore or in the branches form, only one will succeed in reaching the embryo sac. An example of Polysiphonous is Malvasia family or Cucurbitaceae family. We know that style can be hollow or solid. So if the style is hollow, then the pollen tube has got the space to grow. But if the style is solid, then the middle part is filled with the parenchyma tissue, which is also called as conductive tissue. So the middle portion over here you can see is filled with parenchyma tissue, also called as conductive tissue. So over here the pollen tube has to push through the style in order to grow. The conducting tissue has a lubricating material that helps in the movement of the pollen tube, and the pollen tube grow by pushing through these tissues because there is no space. So there is a possibility that the content of the pollen tube might be pushed backward due to the squeezing of the conductive tissue. So as a result, all the content of the pollen may not come along with the pollen tube growth. So to avoid this, there are callous plugs formed at a regular intervals to prevent the backward movement of two male gamete and the tube cells along with its cytoplasm. So over here these are the callous plugs which ensures that the two male gametes and the cytoplasm and the tube cells are at the tip of the pollen tube. And these callous plugs, they are formed at regular intervals. Pollen tube after reaching the ovary enters through the ovule through the micropyle and then enters through one of the synergids, and it is guided, the entry of pollen tube is guided by the filiform apparatus. So all these events right from the deposition of the pollen grain on the stigma until the pollen tube enters the ovule are all together referred to as pollen-pistil interaction. We know that pollen-pistil interaction is a dynamic process. It involves pollen recognition, which is followed by promotion or inhibition of pollen. Now, the knowledge which is gained in this area helps the plant breeder in manipulating pollen-pistil interaction to get the desired hybrid. So, plant breeder, they manipulate the interaction between pollen and pistil in order to get the desired hybrid, and this brings us to our next topic, which is artificial hybridization. Artificial hybridization is one of the major approaches for crop improvement program. That means it is used to improve the quality of the plant by combining the desired characters of the plant. Now, artificial hybridization involves many processes. One of the first step is the selection of the plants. Say for example, if I want a long grain and disease resistance basmati rice in one plant, then I am going to select long grain basmati rice which is long but is not disease resistant, and second plant which is disease resistant but the grains are not long. So first and foremost is the selection of the plant. So whichever characters, desired characters we want, we have to select that plant. So selection of the plant is done manually by looking at the characters of the plant. As we are going to cross the female flower with the desired pollen grain, and in such crossing experiment it is important to ensure that the only desired pollen grain are used for pollination and the stigma is protected from contamination. Contamination means unwanted pollen grain should not land onto the stigma, and this is achieved by emasculation and bagging. First, we are going to study about emasculation. So, the first step was selection of plants. Once the plants are selected, the next step will be emasculation. Now, let us understand emasculation. If the flower is bisexual, then we have to first select the female flower, and the next step is removal of the male flower, that is male reproductive part, and that removal of the male reproductive part or the male flower is known as emasculation. It is required only if the flower is bisexual. That means male and female reproductive part both are present in one flower, that is androecium and gynoecium both are present in one flower, because we don't want self-pollination to take place. And in case of unisexual flower, there is no need of emasculation because the flower is unisexual, so no need of emasculation. Only thing is we have to protect the female flower from unwanted pollination. That's the reason why the female flower bud is bagged before the flower open up. How is emasculation done in the flower? If the flower is big, the filament is trimmed off using the pair of scissors, or the anthers are removed using the forceps, that is manually. And if the flowers are small, then tiny suction pumps are used to remove the anthers. If the anthers are on long filament, then they are dipped in hot ethanol before the maturity of the anther, and that is before the formation of pollen grain. So we have seen that emasculation, it depends upon the type of the flower. If the flower is big, then the anthers are removed using a pair of a scissor or forceps. If the anthers are small, then they are removed using tiny suction pump. And if anthers are on long filament, then they are emasculated by dipping them in hot ethanol. So emasculation depends on the type of flower we are dealing with. The next step is bagging. Emasculated flower must be covered with a bag of suitable size. Generally, the bag is made up of butter paper, and it is to prevent the contamination of stigma with unwanted pollen, that is unwanted pollination should not take place. That's the reason why the emasculated flower must be covered with a bag of suitable size, and this process is known as bagging. So the next step is collection and dusting of the desired pollen grains. The pollen grains from the desired plant is collected by bagging the flower. So once when the anther dehisces, the pollen grains are collected in the bag, and when the stigma of the bagged flower attains the receptivity, the desired pollens are dusted on it. The flower is rebagged and allowed to fertilize, that is fertilization takes place. Formation of seed and fruit takes place. The last step in artificial hybridization is testing of seeds. So the seed thus formed are allowed to grow into a new plant, and hybrid seeds are thus tested, and if we get our desired characters, that means that we have succeeded in the entire process of artificial hybridization. Today we have discussed about pollen-pistil interaction. It begins with pollination and ends with fertilization. The pistil has the ability to recognize and accept the right or the compatible pollen of the same species. The compatible pollen absorb water and nutrients, mainly sugar, through the germ pore and produces pollen tube. Pollen tube reaches the ovary, enters the ovule through the micropyle, and enters one of the synergid through the filiform apparatus. We are going to discuss this in detail in our next video, that is double fertilization. Next, we have studied about artificial hybridization and the steps which involves in artificial hybridization. The first one is selection of the plants, followed by emasculation, bagging, collection and dusting of the desired pollen, rebagging and testing of the hybrid seeds. That's all for today. Thanks for watching this video. If you have any questions, do write to me at alnasacademyofficial@gmail.com. An entire playlist will be uploaded soon. So till we meet next time, stay safe, stay positive, do like, subscribe and share this video with your friends so that they may also benefit. Till we meet next time, take care, bye bye, thank you and happy learning.